In today's second tutorial on the chemistry of medicinal drugs, we will go on with having a closer look to structure and molecular interactions within lipid nanoparticles, which are essential for anti-COVID vaccines based on modified RNA technology. In the first video, we have discussed the overall structure of those nanoparticles according to the result of Professor Tobias Unruh. Well, what we have is a core, an inner shell, and an outer shell. The core has a diameter of 40 nanometers. Then the inner shell, 2 nanometers, it is lipophilic, and the outer shell has 4 nanometers, it is hydrophilic. In the first video, we have already discussed the molecules that are responsible for the formation of the inner and outer shell. Within the core, we have the modified RNA. How does this look like? Well, it is a very long chain of nucleotides, about 4,500 nucleotides, which are connected by this phosphate backbone. We have the classical nucleotides, but in addition, in the modified RNA, we have this so-called N-methyl pseudouridine, which is responsible for a higher stability, for instance, against hydrolysis. Well, as we see here, normally we have a sensitive NO acetal. However, in this case, that nitrogen here is missing. Therefore, this is an ether functionality, and as we know, ethers are far less sensitive against hydrolysis. So overall, this is a highly hydrophilic molecule, and it always carries some water molecules with it. So, what else do we have in the core? The ALC 315. Well, it's this molecule, and very often this molecule is called a cationic lipid. Well, however, there is no cationic charge in it, but it only needs to be protonated at the tertiary amine. Well, then we have an ammonium cation, and this already occurs in the contact with water in equilibrium. Then we have an ammonium hydroxide, of course. So, and this ammonium hydroxide electrostatically will orient to the phosphate backbone, and a rather strong hydrogen bridging bond will be formed. Let us compare this molecule to an octopus. Then we have the octopus mouth biting here at that backbone. We have one polar tentacle with an OH group, also a polar group capable for forming hydrogen bridging bonds somewhere at other polar groups. Then at those tentacles we have, in addition to ester functionalities, we can compare it to tentacle suckers, bipolar interaction, or as hydrogen bridging bond acceptors. Those will also coordinate to polar groups here. So if we have enough of those molecules, and we do have enough, remember we have 430 micrograms of ALC 315 per injection and only 30 micrograms of the Mod-RNA. So we have enough of this molecule to completely wrap that long chain of the RNA. And now those final tentacles are highly lipophilic and hydrophobic. So they will point away from this molecule, will be outside, and the completely wrapped RNA at the outside then is highly lipophilic due to those hydrocarbon tentacles. Well, this fits to the overall lipophilic character of that nanoparticle core with cholesterol in addition in there, mainly also lipophilic. And then the lipophilic inner shell fits to that core, and the hydrophilic outer shell then is responsible that the whole assembly is soluble in water. Thanks for listening.